So, good afternoon and a very warm welcome to all of you, and thank you for joining us for today's webinar entitled Rapid and Reliable Detection of Medulloblastoma-Associated DNA Methylation Patterns, MS Mimic, organized by the Journal of Precision Medicine and proudly sponsored by Agena Biosciences. Now, Agena Bioscience is dedicated to advancing the impact of genomics in healthcare and precision medicine. Their highly sensitive and cost-effective mass spectrometry-based platform, the mass array system, is used globally in diverse fields such as cancer profiling for solid tumors and liquid biopsies, inherited genetic disease testing, pharmacogenetics, agricultural genomics, and clinical research. Their mission is to equip genomic and clinical testing laboratories with practical solutions that can increase productivity and decrease time to results, whether assessing sample quality, screening samples for actionable mutations, or enabling routine genetic testing for tens to thousands of samples. Their products and services help laboratories translate genomic discoveries into mainstream clinical practice. Now, I'm Damien Doherty. I'm the Editor-in-Chief of the Journal of Precision Medicine. I should be moderating the webinar this afternoon. I would encourage you all to send your questions to the speaker throughout the presentation using the function you'll see on the dashboard. We'll, of course, do our utmost to answer as many of these as we can during the Q&A session at the end. Okay, so without further ado, let me introduce our speaker today. Ed Schwalbe is a senior lecturer in bioinformatics and biostatistics at Northumbria University, Newcastle, and has worked on childhood brain tumor medulloblastoma for more than 10 years. He led the development of the MS Mimic assay for the routine assessment of diagnostic DNA methylation signatures, and his research interests include the development of translational assays that bring research findings into routine clinical use. Ed will be discussing how the detection of disease-associated variation in DNA methylation profiles holds significant potential for diagnostic and research applications. However, sparse and degraded samples often pose a challenge on a number of technologies. Ed will discuss how their solution, MS Mimic, will rapidly and reliably detect medulloblastoma-associated DNA methylation patterns in pediatric cases. So, Ed, the floor is yours, and we look forward to listening to your talk.
Okay, well, thank you, Damien, and hello to everybody who's listening, and thank you to the Journal of Precision Medicine for giving me the opportunity to speak today about this assay. So just before we start, my email address is there on the first slide, so if you have any questions that are not answered during this webinar, please direct your questions to me, and I'll do my best to answer them for you. So the MS Mimic in the title there stands for Mass Spec Minimal Methylation in Cancer. So that was the acronym that we chose for the development of this assay. So I think with the advent of genome-wide methods to assess DNA methylation at a relatively low cost, so I'm thinking here about the Illumina methylation arrays, it's become very clear, and there's a growing body of research that describes aberrant DNA methylation and how that is a characteristic recurrent feature of human disease. So, as Damien mentioned, I'm going to describe this MIMIC assay, and it specifically was designed to be both rapid and low cost, but also very robust, and this is for the assessment of DNA methylation signatures. So these diseases are being shown to have methylation signatures which distinguish particular variants of a particular disease. So in particular cancer, can you identify a minimal signature and then use a PCR-based approach suitable for analyzing very scant and very degraded samples to assign subgroup in a way that is clinically applicable. So in order to be clinically applicable, it needs, particularly for the brain tumor that I work on, which we'll come on to in a second, it needs to be applicable to single samples because it's a relatively rare disease, but also have the scalability to move to high throughputs if necessary. And in particular, it needs to be able to be turned around rapidly. It needs to work with poor quality materials. So we were thinking about formalin-fixed, paraffin-embedded materials that are typically collected in routine pathology, and it needs to be low cost. So we've developed this assay. We've used this brain tumor, medulloblastoma, as an exemplar disease, and we think it has two major applications, both of which I'm going to talk about in this webinar. So the first of these is to unlock archival cohorts. So many archival cohorts will be large hospital collections that are paraffin blocks that may not have enough quantity or quality of DNA for gold standard approaches such as DNA methylation arrays or whole genome bisulfite sequencing. This minimal signature here works with very small amounts of DNA, so this has the potential to unlock cohorts that were previously refractory to modern genomic analysis. Also, it has the potential for diagnostic applications. So we're seeing a lot of advances in the research side of this disease, medulloblastoma, but what is needed is the transfer of these research findings into clinically applicable assays, and I'm going to talk about how we've implemented this here in the UK in Newcastle. Okay, so I think it's best to talk about this disease. It's not necessarily a disease that many of you will have heard of. It's a paediatric brain tumor of childhood, and you can see here on the MRI scan, this white mass that I'm indicating with the highlighter there is the tumor medulloblastoma, and it's presenting in the cerebellum. So gratifyingly, it is rare. So just to put it into context, there are between about 80 cases a year in the UK and 350 cases a year in the USA. However, it is a disease with devastating consequences. So the primary treatment modality is surgery. So neurosurgeons will come along and chop out as best they can the maximal amount of medulloblastoma tissue there. And then after about a 4-week window, so the patient has 4 weeks to recover because this is obviously quite an invasive and major operation. After 4 weeks, the patient will be commenced with chemotherapy and or radiotherapy. And those choices of chemo and radiotherapy are informed by currently understood risk factors. So at the moment, children aged under 3 are not given radiotherapy routinely, simply because this is simply too devastating for their developing brains. If the tumor has metastasized as well, then the patient will be treated with more aggressive protocols. And also if the tumor was incompletely resected. So if the tumor happens to be infiltrative and is difficult to completely resect, then the patient is also classified as high risk. So on the face of it, the 5-year survival, which is between 70 and 75% for this disease, is relatively encouraging, particularly because survival back in the 1970s was about 2%. However, there are undesirable side effects of treatment. So patients, survivors of medulloblastoma will have lifelong, life-limiting effects from their treatment. So they will find it difficult to have a job. They'll have psychosocial issues, neuroendocrine defects, will be unable to have all of the things that we would take for granted as part of the sort of trappings of becoming an adult. So what's really needed is for an improved subclassification of the disease. So about 10 years ago now, people started taking this disease, which was previously thought of as a single entity, and applying transcriptomics and DNA methylomics to this disease, and it became very clear that there were molecular subtypes of this disease, and that's what this diagram here is alluding to. So there were various publications starting in 2006, and eventually the field reached a consensus in 2012 in this consensus paper here. And what it is saying is that there are 4 molecular subgroups of this disease. So starting with the first one there, Wnt is a tumor characterized by inappropriate activation of the Wnt-Winkler's signaling pathway. And the SHH group there in red is characterized by inappropriate activation of the sonic hedgehog signaling pathway. And then thereafter, we have 2 other groups with generic names, and they have those generic names because the trigger or the driver of tumorigenesis in these tumors is still poorly understood. So what's important clinically of these subgroups is if you look at the prognosis highlighted there, the Wnt group has a very good survival. So these patients with a Wnt subgroup tumor will have about a 95% survival. So that's obviously very good. However, patients who are group 3 have a relatively poor survival, so that's about 60%. So what we need is the ability to undertake upfront molecular subgrouping of these tumors in a way that this can be used to inform treatment decisions. So really, we're thinking about risk-adapted therapeutic stratification of these patients and the ability to place patients onto the appropriate treatment protocols. So these would be aimed at therapy reduction, for example, in the Wnt patients, where we would expect the patients to survive, and also intensification of treatments if a patient happened to be group 3. So the long-term goal would be to spare patients with a good prognosis the full extent of the treatment and to give them a better quality of life post-survival, and also attempting to continue to aggressively treat those patients with a poor prognosis. So moving on, and bear with me. Yep. So we were the first group back in 2013 to describe that you can identify these molecular subgroups, sonic hedgehog, group 3, Wnt, and group 4, using DNA methylation patterns. So we did this with first generation Illumina Golden Gate methylation arrays, and the V1 through V4 that you see here are metagenes. So metagenes here are the combination of many hundreds of probes which are joined together to describe the major biological effects within a data set. So that was the first demonstration that you could identify methylomic subgroups of medulloblastoma, which corresponded very nicely to the Wnt, sonic hedgehog group 3 and group 4 subgroups. So this work, that the development of this assay was funded by a large UK charity, Cancer Research UK. It was entitled Biomarker-Driven Individualization of Medulloblastoma Therapy. So essentially that involved us profiling many hundreds of tumors with RNA-seq to look at gene expression patterns, Illumina 450K methylation arrays to look at genome-wide methylation patterns, SNP arrays, and also targeted sequencing to identify specific hotspot genes for mutations. And the work fell into this work package 3. So what we said was, yes, we're going to identify interesting disease biomarkers, but furthermore, we're going to develop these into clinically applicable assays. So this was the problem that we were facing. Many different methods have been described or published to identify the molecular subgroups, but all of them have limitations with regard to their application to routine use. So, for example, the methylation array has to run in multiples of 12. So for a rare disease like medulloblastoma, that can be problematic. However, the methylation arrays still remain the gold standard. So what we wanted was to think about a clinically applicable assay for molecular subgrouping of medulloblastoma that could be used routinely. So we thought about what might make an ideal medulloblastoma subgroup assay. So of course it should be inexpensive. So I'm sure in the UK, as well as in many other countries, healthcare doesn't have a bottomless pot of money. So it needs to be compatible with NHS funding in the UK. It needs to be rapid. So that was thinking about the 28-day turnaround between removal of the tumor and the commencement of appropriate therapies. So 28 days might sound like quite a lot, but with a rare disease, this is presenting in hospitals all over the UK, and the samples would then have to be shipped to Newcastle, would undertake DNA extraction, etc., run the assay, and then communicate it back. So it can be quite tight to turn that around within 28 days. This was another major thing, was that, as I've mentioned before, it had to be compatible with formalin-fixed, paraffin-embedded standard pathology samples. It had to be applicable to singleton samples in the context of this rare disease, and we were keen that the assay should be transferable. So while we could offer this as a service in Newcastle, any other laboratory that wanted to implement this assay in their own labs would be welcome to use our expertise and our assay for that. It also had to have quality assurance in terms of robust QC measures. And then finally, yes, it also had to be robust and be reliable. So we looked around, and there was no real existing off-the-shelf solution that could answer all of our needs. But what we did see, I do see that it says sequinome there, but it's now Agena, of course, is to adapt the Agena IPlex SNP assay to assess DNA methylation in multiplex. Okay, so I'll come on to the mechanics of how that assay works in a few slides' time. So, as a result of our Cancer Research UK program grant, we had a large cohort of 220 primary medulloblastoma tumours that have been run on this Illumina methylation array here. So you can see that it runs in, each sample would run in one of these wells here. So there are 12 on an array. This has been a very versatile platform in our hands. It assays CpG loci at 475,000 positions across the genome. It's now been superseded by its successor, the EPIC array, which has about 850,000 loci assayed. So this represents, of course, a rich source of potential biomarkers, and it really can be considered, I think, genome-wide, in that 99% of genes have at least one probe targeting them, and it covers 96% of CpG islands. So although it's very robust and gives a large amount of information, what we were hampered by was the amounts of DNA required as starting material. So Illumina would recommend you work with 1 microgram of DNA as your starting material, although you can push it down to about 200 nanograms. And with the cohorts we were working with for this assay, this just was not possible. So as part of our program grant as well, we've run the same molecular subtyping assays on the molecular subtyping studies on the 450K array, and again we found 4 major subgroups in a cohort of 428 primary medulloblastomas. So just briefly, we see the Wince subgroup here, the sonic hedgehog subgroup here. This is the group 4, the shades of green, and the yellowy orange shades is group 3. And again, we've identified subtype defining metagenes, so combinations of methylated loci that define the major biological effects within our data set. So we knew that we could identify these groups using the 450K array platform, but now we needed to turn this into an assay that would work for us. So this is the assay, and it was published in 2017 in October, so relatively recently, although of course the work that went into the production of this took several years. So I mentioned before that we wanted to adapt the Agena IPlex assay to measure DNA methylation patterns in multiplex. So Agena already offered the EpiTyper assay, which would measure a single stretch of a few hundred base pairs and assay every single CpG dinucleotide within that region. So what we wanted here was a more bespoke approach that could assay individual CpG loci at multiple points across the genome. So we thought that we could identify a methylation signature which could define the molecular subgroups and then interrogate that using this multiplex PCR-based approach. So just to take you through how this works, at the top here we have a biomarker defined by DNA methylation, and on the left-hand side the red M's there indicate a methylated cytosine, and on the right-hand side the lack of M's indicate an unmethylated cytosine. So all of these assays, the 450K array and also this assay, rely on sodium bisulfite conversion. So what you do is you treat your DNA sample with sodium bisulfite. Methylated cytosines will remain as cytosines, whereas unmethylated cytosines will be deaminated into uracils. Okay, so that in effect creates a methylation-dependent SNP. So you can assay a particular signature of methylation-dependent SNPs that you've created, then you'll be able to start to identify these molecular subgroups. So essentially, you then carry out a PCR, a multiplex PCR of this bisulfite-converted DNA, and the uracils will be read as T's when you carry out the PCR. And then it feeds into the standard IPlex type of assay, in that you anneal an extension primer adjacent to the cytosine of interest, and then you have a single base extension. You either add a G, as shown on the left-hand side here, indicating a methylated cytosine, or you add an A, as shown on the right-hand side here, indicating an unmethylated cytosine. So you have those reactions, and then you can put those onto the MALDI-TOF Agena mass array, and you can look at the pattern that you get there and quantify particular peak heights with particular variants. And of course, because you're doing this in multiplex, you can assay many loci at the same time. So that was the assay, at least in principle, and what we really needed to know was whether or not we could actually do this in practice. So I'm going to describe a little bit about the development of the assay and then how we've tested it and implemented it on, firstly, archival cohorts and then routine use. So this is where we started. We had a cohort at the time of 220 medulloblastomas. We used, as it says there, NMF clustering. So this stands for non-negative matrix factorization clustering, but it's what I've been describing on previous slides. We pulled out the major biological effects within the dataset and described them as metagenes. So that's shown at the bottom here. You can see that each of these molecular subgroups, sonic, Wnt, group 3 and group 4, are characterized by strong expression of this particular metagene. Okay, and you can also see this data represented in another couple of ways here. This is a principal component analysis plot here, which shows the variation within the data set, and I've drawn spheroids around the molecular subgroups as determined by us, and you can see it falls fairly nicely into discrete subgroups. You might notice there's a slight overlap between groups 3 and 4 there, which are shown in yellow and green respectively, and that's definitely a feature of the disease. There's clear evidence that group 3 and group 4 have some interrelation. One of the ways you can assess the quality of your clustering is by looking at silhouette plots. And what we can see here is that every sample appears to be well clustered, and that is shown, that the evidence for that is that each sample, which is shown as a row in this silhouette plot here, has a positive silhouette score. So if a sample were incorrectly clustered, it would have a bar going off down into the negative territory here, and that's what we don't see. So we had a cohort of 220 medulloblastomas run on the 450K methylation array that we were fairly happy were representative of the disease and from which we could pull out 4 molecular subgroups. Okay, so the next part was to identify subgroup methylation biomarkers. So biomarkers or loci in the genome which are specific for a particular subgroup. So to do this, firstly, we identified the 50 loci or 50 probes that were most significantly associated with that particular subgroup, and then went through a primer design process. So firstly, you had to determine whether or not the locus that you'd identified was amenable to primer design. So just to go back a couple of slides, and bear with me here. Of course, when you do this bisulfite conversion of your DNA, you change the primary sequence. So it's not just straightforward. It's not just as straightforward as designing regular primers. You have to account for changes to the sequence that flanks your CpG of interest. So you have to approach that with caution. So first of all, you have to make sure that the primers are suitable for assessment, and you can the Agena software can help identify primers that may be suitable for this. Then you have to assess whether they're compatible in multiplex as well. So it's possible, of course, to run, in this case, 17 singlet reactions, but we want to reduce this, the amounts of PCR we do as much as possible, so it'd be advantageous to multiplex these primer sets together so that you can assess multiple loci in the same PCR reaction. So there's a long and reiterative process of testing particular primer combinations, checking that they worked singly and also in multiplex, and before we put them all together. We also had to design a classifier based on the selected loci, and we wanted to do this in such a way that you could include some degree of redundancy within this assay. So what I mean by that is we were conscious of the fact that we were working with potentially very degraded DNA. So it was highly likely that in any given sample, you may get one or more loci dropping out simply because they were not accessible due to the degradation of the DNA. So therefore, we wanted to ensure that if a particular locus did drop redundancy to overcome that missing data and still produce a subtype call. And later on, I'll talk about the empirical determinations of quality control that we implemented. So that we had 17 loci, and their Illumina identified alongside their chromosomal position. And the heat map shows the DNA methylation. So here, a blue signal would indicate low methylation, and a red color would indicate high methylation. So you can see few selected, bear with me, my laser pointer is a bit slow. The first few probes selected here are picking out the sonic hedgehog subgroup of medulloblastoma. There's methylated there. And the probes here, for example, are picking out the Wnt subgroup. So with these generation of these multiplexes, eventually after 2 rounds of optimization, we identified 17 loci, which were split across 3 multiplexes. So the Agena design software identifies which primer sets are likely to work in multiplex, and that's what we are working from. So in this case, from these 17 loci, we'd identified 3 multiplexes. Okay, so what you're looking at is the x-axis true methylated DNA. So Dr. Henning Golka, who worked for Agena, mixed unmethylated and constitutively methylated DNA in 10% increments, which is shown along the x-axis, and then assessed the ability of the assay to predict the percentage methylation on the y-axis. So what you're looking for is this kind of linear relationship that you see for all of the assessed loci. 1.8, which hopefully shortly, yep, here we go, which is flat, and this is an additional control that we built in to ensure that complete bisulfite conversion was achieved. So alongside this process, we also had in mind working with degraded. Of course, we wanted to our amplicon as small as possible. So in this case, amplicon 17 loci from about 50 to 100 base pairs. So we thought that that would maximize our chances of success. And just again, to go back one slide, what I forgot to mention about this heat map here was that hemimethylated loci would be shown in white. So you can see that generally there's an absence of hemimethylated loci here, and we tend to see methylation at either extreme of the either being completely unmethylated, i.e. wholly blue, or completely methylated, wholly red. So what we wanted to do was to maximize our chance of being able to separate these biomarkers. And I think if anyone was considering doing this for their own assays, that would be something that would be quite important, would be to ensure that you get a large change in methylation when you're designing these or identifying these biomarkers. So on top of the assay linearity, of course, which was great to see, what we wanted to do was assess concordance with the gold standard, which is the 450K methylation array. So the beta value there is just the fraction of methylation at a particular locus. So more densely populated regions are colored red, and the sparse regions are colored blue there. So what ideally you'd be seeing is a line across the diagonal here, which is typically what we see, but also see the point that I was about, the loci that we identified being at either extreme, either completely unmethylated down here or completely methylated. So we identified a good correlation between the methylation array and the estimates that were made by the MS mimic assay. So the assay itself is fairly straightforward. You extract DNA. We quantitated DNA, and we didn't rely on nanodrop. We used qubits, which is a way to assess specifically double-stranded DNA, as we felt that this gave a better overall handle on the quality and the amount of DNA that you had. Then we carried out a bisulfite conversion. So if it were available, we would convert 100 nanograms, although we worked with anything that we could get our hands on. and talk about that in a few moments. We carried out a quality control PCR. So this was a PCR designed to work on bisulfite treated DNA that would amplify a target of 200 base pairs. So the idea being, of course, to amplify a 200 base pair fragment, then in the multiplex where we're looking at fragments from 50 to 150 base pairs, then that should be more than sufficient. So then we carried out the multiplex PCRs and then transported the PCR product to Newgene, which is a diagnostics company based in Newcastle. It's a venture between the National Service, the NHS, and Newcastle University, and it offers routine genetic testing. And one of the platforms that they have available is the Agena assay. So the Bioscience identify the cases and do the processing. So that was development. We've handed the entire assay over to Newgene after successful validation. Okay, so we designed the biomarkers, identified the biomarkers on a cohort of 220 medulloblastomas. Now we took an additional independent cohort of an additional 106 samples, which we called the gold cohort, representing all subgroups, Wnt, Sonic, group 3, 4, and also representing a variety of different qualities. So of course we started DNA extracted from fresh frozen tumors, because of course if you doesn't work with that, then you're in big trouble. And then we started adding progressively more challenging samples, formalin fixed embedded DNA, and then last cytospins. So if anyone's unfamiliar with what a cytospin is, this is one of the applications we've envisaged. We're applying it to a clinical cohort, which was called PNET4. So not enough material remained to assess this using conventional genome-wide techniques, such as the 450K array. But what was left over from this trial was cytospins. So this is where you've undertaken a nuclear extraction of DNA, spun those nuclei onto a glass slide, and typically you'd use them for fish. So in our disease, we look for amplification of specific oncogenes, so specifically MYC and MYCN, which are very powerful oncogenes, and you can identify them using fish. So the pictures at the bottom just show what we're looking for. So what we do is circumscribe the nuclei with a wax pencil, and this would act as a sort of reservoir for any lysis buffer that you might wish to add to your glass slide. And you might be able to see the nuclei there at ×10 as the white dots. And then you would literally take a scalpel and scrape across the slide and hopefully remove all those nuclei post-extraction, and you would carry out the assay from what you scraped off this glass slide. So this really did represent the dregs in terms of what we could reasonably expect to apply to this assay. These have been kept for 10 to 14 years in storage, because the assay ran from 2001 to 2006, just kept at room temperature, so it really was a potentially challenging cohort to work with. As I said, there were 106 samples for this validation cohort. We ran the assay on this, applied the classifier that we developed on these 17 loci that we'd identified, and then empirically determined appropriate QC measures. So how would we decide whether or not the assay had worked? Can we give a reliable subgroup call that we're happy with? Because obviously with the intended application of this, it's of fundamental importance that you make the right call in terms of assigning a patient to a particular subgroup. So you cannot get that wrong. And obviously we want to be conservative when making that. So we were right from the start very happy to have a classification of unclassifiable in order to try and prevent misclassification. Okay, so we didn't do this routinely, but you can see this is what happens if you run the results of the multiplexed bisulfite PCR out on an agarose gel. You can see the sample sizes there from about 50 to 150 base pairs across the 3 multiplexes. So initially in the early stages, at least, it was reassuring to see that you could actually identify these as bands on a gel. So one of the problems or potential pitfalls is the potential for locus dropout. So if you are working with degraded samples, what do you do with that? So in those 106 samples there, you can see the distribution of missing loci that we had. You can see that the majority of assessed cases had 0 missing loci, and a handful of cases had greater than 6 missing loci. So what we wanted to do was to model what would happen if we imputed this missing data. So imputation is using computers to look at similarly to look at samples with similar methylation, and then to assess what their likely methylation score was for those missing loci. So we tested using this multiple imputation how many loci could be replaced, how many missing loci could be replaced before the performance of the assay started to break down. And to make it really difficult for ourselves, we replaced that missing data with the wrong value. So what I mean by that was, if in reality the methylation score was 0, for that missing data we replaced it with a methylation score of 1. So we wanted to try and account for the potential confounding that could come in by using this imputation-based approach. But what we found anyway was that you could tolerate up to 6 missing loci before assay performance started to degrade unacceptably. And that is what we used moving forward from that. So we had this 101-member validation cohorts, and as I said, we've all of those validation cohort had also been run on the 450K methylation array. So we had a classifier based on the 450K array. We had a classifier based on the 17 loci that were being used in this signature, and obviously what we wanted to do was to compare the classifications made using the gold standard classifier, i.e. this blue one here versus the one based on this minimal methylation signature. So as you may well expect, the assay performed best on fresh frozen derived materials, so DNA extracted from fresh frozen materials. We had 39 out of 40 samples working and giving a probability score for a subgroup assignment above this threshold here. I'll talk about this in a moment. Formalin fixed ran fairly well, so there were 3 failures there, and we had 2 failures in our albeit smaller cytospin derived cohort. So with all of this, though, we were gratified to see that in the main the assay worked and it returned a subgroup call. So we looked at the distribution of the probability of subgroup calls that you see on these 3 graphs here, and we fitted a line to that, and we said that, okay, 95% of samples are falling above or to the right of this blue line here, which corresponds to a value of 0.69. So we said if the probability of assignment exceeds 0.69, then we will regard that as a confident call. Below that, we will say that the sample was non-classifiable. So when we look at this confusion matrix here, which in columns is the gold standard reference subgroup, and in rows is the subgroup predicted by the mimic assay, you can see that there's complete concordance, which was great to see, albeit we did have some non-classifiable samples, these NC samples here. So we'd satisfied ourselves that the assay was robust. It worked on an entirely independent cohort from the one that we had developed the initial signature on, and seemed to be working reproducibly and robustly. So now I'm going to talk the first application that we put this to, which is the methylation analysis of archival cohorts. So as I already said, the assay has low sample requirements, so less than 100 nanograms is suitable. It works with very poor quality DNA, so thinking about the cytospin extracted DNA there. And what we think is that it might have applicability across many diseases and many types of cohorts, which were previously inaccessible using conventional techniques. And yeah, so what I'm going to talk to you about is the analysis of pre-omics era cohorts. So this, I think, is nodding to older cohorts whereby collection of high quality material wasn't mandated, and retention of biological material was also not mandated. So you have to work with what you can get, which can be a very small amounts. So the clinical trial that we had was HIT-SIOP PNET4. So with a rare disease like medulloblastoma, it's typically run with trials run across Europe, and there'll be concurrent trials, albeit with a different purpose, running in North America. So the idea is that if anything interesting comes out from either the European or North American trials, then it can be validated in the others cohort, and then that can be fed into new clinical practice. So this ran from 2001 to 2006, so predated the discovery of these molecular heterogeneity within medulloblastoma, and as a consequence, there was no provision for collection of materials for biological analysis. So what we had was what I've previously shown you, the cytospin slides left over from fish. So the original medical study was published here. So the purpose of the trial was to compare different methods of radiotherapy. And then this is the biological trial, which was published a couple of years ago now. And we had a lot of trouble publishing this study. And the big criticism that we had from reviewers was that it was unacceptable in this sort of subgrouped era of medulloblastoma for us not to have included molecular subgrouping of our cohorts. And we of course, we tried to explain that there simply wasn't material for that, but that didn't really cut much ice with the reviewers. So eventually we published it in OncoTarget, but of course the study would have been a lot stronger had we been able to identify molecular subgroups. So that was one of the driving forces behind the development of this assay. So this is the flow chart of how the assay worked there. So of the 338 cases that made the original study, we had material available of one sort or another from 161. We extracted DNA where we could. None of these extractions had sufficient material for conventional 450K or EPIC methylation array analysis. And then from that, we found that 153 of those was potentially suitable for this assay. 107 of those 153 passed the assay quality control measures, and then we were able to look in a subgroup dependent way about survival and the associations with clinico-pathological correlates in this clinical trial. So this is the application of the assay to this PNET4 clinical trial. Each one of these dots represents an individual sample, and they've been ordered in sort of ascending probability. So you can see that a large proportion of those than perhaps we'd seen before were unclassifiable, but nevertheless, I think we got information for about 107 of those samples. It's really striking, I think, that the assay performed well, seemingly regardless of the amount of input DNA. So although nearly all the samples worked if we had greater than 80 nanograms of input DNA, we still had a fairly good 2/3 response rate or 2/3 success rate for samples where there were fewer than 2 nanograms of DNA available. And this is, of course, the level of detection of the cubelet fluorimetry that we were using. So it was below 2 nanograms, but yet we still got a result for many of these samples. So there's the reasons and the breakdown of where we had assay failure. And of course, this is probably the most challenging of challenging cohorts. So we were relatively happy about the success rate here. And what it enabled us to do, so this PNET4 trial was a trial of standard risk medulloblastoma. So these are tumors which are absent for currently understood high-risk disease features. So they are non-metastatic, they are children aged over 3, they are completely resected by the neurosurgeons. And what we could see is that this is a Kaplan-Meier curve for anybody who's not familiar with looking at this type of thing. It's very commonly used in cancer, and it shows the proportion of patients in particular subgroups surviving over time. So you can see that if a patient is going to relapse, then it's going to happen typically within the first 5 years, and that's what we expect to see in the disease. And what we see here is that the group 4 samples harbor the most disease relapses. And unfortunately, when a child relapses with medulloblastoma, there's nothing really more that can be done. You only get one chance to apply radiotherapy, and then you just have to palliate after that. So it's important to know the underlying molecular subgroup behind this, and using this MS mimic assay, we were able to identify that to unlock archival cohorts. And we have this finding that standard risk group 4 medulloblastoma is associated with worse outcomes. And we're currently preparing another manuscript which goes into this in more detail. So the MIMIC assay here has been fundamental to the creation of this subsequent study. So the other application of this MIMIC assay is adoption into routine clinical practice. So we were keen that this could translate from our research setting, which I guess is the PNET4 work that I've just described to you, into a more routine setting. So this involved transferring the assay in its entirety to Nugene. We've run now 160 diagnostic UK cases. So 50 of them have come from us in Newcastle, and 110 have come from Great Ormond Street. So many of you may have heard of Great Ormond Street, which is sort of the major children's hospital in London. So the assay turnaround time, or the mean turnaround time, is 10.7 days. We get about a 15% failure rate overall. So this is perhaps a little bit higher than we'd observed when we were carrying out the assay in our own hands. But I think we also have to be aware that when we were doing it in our own hands, we were frequently, if something failed, we'd have a second go. We could probably curate these samples a little more. They weren't just forming part of a routine workflow. So the assay seems to be working well. It's being used now as a standard diagnostic test in the PNET5 clinical trial. So this is really the first implementation of what I mentioned earlier. So we can upfront identify this WNT subgroup of patients, treat them with less aggressive therapies, and then in 5 years' time, we'll be able to assess whether these patients have an improved quality of life post-survival. We'll also be able to see whether this reduction in intensity of therapies has not had an adverse impact on the relapse rate as well. So it's going to be very interesting to see in a few years whether this therapy de-escalation has been effective or has worked in these patients. So you can identify WNT and sonic hedgehog subgroup with these two antibodies here, and we see agreement there where possible. So we do see a failure rate, but it's not too dissimilar with the failure rate that we see, for example, using routine FISH. So FISH is routinely done in the NHS for the amplification of these oncogenes, which identify high-risk patients. So now this is being used for UK cases in the PNET5 clinical trial, and it's feeding forward to clinicians and can be used to assign patients to a particular treatment. So coming to a close now, I've described this MIMIC assay for subgroup assignment in medulloblastoma. It seems to work reliably and rapidly and robustly from 100 nanograms or less of DNA. It's relatively inexpensive as well. So you can work to good GCLP at this commercial provider, and it's relatively inexpensive. It has a fast turnaround. It can be adopted and transferred to commercial providers of the Agena platform. And we have put or made available open source algorithms for the sample assignment and made a web classifier so that Nugene, the company that runs the assay, can feed the outputs from the machine from the mass array mass spec machine onto this web server, and it will carry out automatic classification. So it will give a subgroup call and the confidence with which that call has been made, and you can also show this graphically in a similar way to what we've discussed before. So I think, of course, this is a very niche application that I've been described to you, and we had a sort of very specific use case when we were trying to implement this, but I think it does have a wider significance. So in particular, pediatric cancers, brain tumors, leukemias seem to have very tightly defined DNA methylation signatures. So there's a clear application there to identifying molecular or methylation-dependent subgroups in other tumors, particularly pediatric tumors, but I think it also has some application to adult tumors. Just outside of the sort of molecular subgrouping, though, as well, you can identify methylation biomarkers that might mean something in terms of susceptibility to particular disease, or, for example, at the bottom right there, association of epigenetic signatures with BMI is the sort of things that you could derive signatures for and then apply them at a low cost in very large cohorts. I think also you could apply this in a non-invasive way. So liquid biopsies is becoming an area of great interest. either in the blood or in CSF. So, of course, with this brain tumor, it's still a relatively unanswered question as to whether you can identify tumor-specific DNA in blood. But I think the detection of DNA methylation patterns has advantages over detection of specific mutations, because you can capture a much larger proportion of a particular tumor type by its methylation signatures rather than by looking at specific mutations. So this is just looking at towards that. So one of the work packages that we're working on is in development of less invasive biomarker assays. One of the techniques we're using is magnetic resonance spectroscopy, but one other potential avenue for exploration would be to see whether we could identify tumor-specific DNA methylation patterns in cerebrospinal fluid. So survivors will have routine lumbar punctures, and we can collect some of that for research purposes and see whether we could identify tumor methylation patterns as a precursor for detecting tumor relapse. It might be more sensitive than what is currently done. And so that's thinking about the longitudinal monitoring of CNS relapses. In a preoperative sense, potentially you could identify the molecular subgroup before surgery. There are sonic hedgehog pathway inhibitors available, so you could potentially shrink a tumor prior to surgical excision using something like vismodegib, which is a smoothened inhibitor. You could perhaps, if a tumor has a good prognosis and you know this up front, you could perhaps less aggressively resect that tumor, and that might spare healthy brain tissue. So I think in the interest of time, I'll just move straight on to the acknowledgements and thank the large team that helped to create this assay and its eventual publication, and also thank you to the Journal of Precision Medicine and Agena for supporting me in this research. Thank you.
Thank you, Ed, for a very interesting talk, a very comprehensive talk as well. So, so bravo there. That was very, very stimulating. We have had some questions come through, so I think we're ready now to start our Q&A session. Thank you for all of those questions that have come in the last 45 minutes. I'm grateful to all of you that have taken the time to submit those. So the first question we have, Ed, is does this technique work for finding methylation in non-disease cells, perhaps to follow inherited methylation patterns?
So, yeah, of course, there's no reason why you should confine this to diseased cells. You can identify methylation patterns in normal, healthy cells. I guess one of the questions you'd have to ask was, what would be, is there any contamination? Are you looking at a completely healthy individual? And if you do detect an aberrant methylation pattern, then what is that a signifier of?
Okay, all right, lovely, thank you. And another one that came in, it says, can you remind us how low a DNA concentration you could go to, any LOD studies?
Well, I can only answer that with referring to what I've already said. So when we were running this, we had the qubit, which could go down to a limited detection of 2 nanograms per microliter. And then we could routinely get samples working below that technique. And we perhaps should have done, but never did get around to sort of doing dilution series to see at what point the assay started to break down. But certainly it seemed to work with very small input amounts of DNA.
Okay, brilliant. Thanks, Ed. Another one here is, what is the upper limit of multiplexing with this approach? Yeah,
so if you run the typical Agena iPlex SNP assay, so on normal DNA, then you can multiplex up to, I think, about 40 loci. So we were very conscious of the fact that when you treat with sodium bisulfite, you reduce the complexity of the DNA. Essentially, you've reduced the 4-base genome down to 3 bases. And there's a lot of potential for secondary structure to get in the way of hybridizing primer sequences to your template strand. So we've pushed it up to sort of between 10 to 15 we thought would be a realistic maximum for this, and then gradually whittled away loci from that, which is why we ended up with 8 in the first multiplex there. So yeah, originally it was working with maybe 12 to 13 loci was achievable. I think no more than about 15 would be realistic.
Okay, all right, great, thank you. Which probably leads similarly into the next question, that it says here, there is often some consideration needed to work with bisulfite converted samples with next-gen sequencing, i.e. balancing nucleotides. Are there similar considerations with this approach?
I don't think so, because you do all the hard work up front. So you identify the loci that you're interested in, and then you validate them using PCR. So in essence, you know that the loci that you're interested in work before you then carry out the assay. So you identify potential candidates for your signature, you validate them in vitro using PCR, and then you can then just run them on samples, and they typically work at that point.
Okay, great, thank you. Another one that's just come in, is this approach currently being used in other types of cancer? I know you touched upon that a little bit at the end there, but I know it can have other applications, but do you know if this approach is being used elsewhere?
So I'm certainly aware of other groups that are using this in the development of particular assays. We published this in October, so I guess it would be too early to say. I'm not aware of any other publications that have using this kind of multiplex signature for methylation assignment, but I think the approach is relatively straightforward and is definitely applicable to many other disease types, or it just not least with the application to cancer.
Okay, brilliant. Thanks, Ed. Another one here. What QC measures are needed to be in place before processing FFPE samples?
So again, we had, I guess, fairly crude QC measures, but it did seem to work. So the main QC measure was, after bisulfite conversion, could you amplify a test PCR of 200 base pairs? And then because we'd ensured that our amplicon sizes were below that, then we were fairly confident that you'd be able to amplify the loci given that 200 base pair PCR was working.
Okay, great. I think we've probably got time for one more. Can you please tell me more about what is needed to analyze the data? What bioinformatics pipelines are needed?
So you get the raw peak height data from coming off the mass array. We used the programming statistical language R to implement both the signature identification, the classifier development, and also the web server, which I showed you the screenshots of, are all running R. The pipelines are, again, relatively straightforward, and be happy to share with anybody interested who might wish to run these sorts of assays themselves. We used an R extension called Shiny, which is able to integrate inputs from the user, i.e. on a web server, and then apply that to their own data. So what NewGene will do will be to run this on the web server, and then they will forward the results to us. So it's entirely hands-off from our point of view.
Okay, great. And actually, I said one more, but there's just one that's popped in, and I feel like it would be remiss of me not to ask it. So can you delineate the subgroups within the subgroups by methylation status? Can this assay assess copy number variants like MYC, MYN amplification?
So yeah, so MYC and MYC-N amplification would not be accessible using this technique. You can infer them using the 450K or EPIC methylation arrays by looking at the intensity of the signals that you get at these MYC and MYC-N loci. Diagnostically, we use FISH to identify MYC and MYC-N amplification. And although at the moment the subgroups within subgroups, so perhaps the questioner is alluding to the fact that recently there have been 3 publications which further subdivide group 3, group 4, and sonic hedgehog. And yes, you could extend this assay to include more loci to identify those, but I think what we'd be waiting for at the moment is for the field again to reach consensus. So we published that there were 7 subgroups of the disease recently. Other groups in Heidelberg and Toronto described 8 and 12 variants of the disease. So the next step, and is what we're currently working on as well with those groups, is to reach a consensus, and then we can start to think about delineating subgroups within subgroups. But yes, it's certainly possible.
Fantastic. Well, that's great. Well, look, Ed, thank you for answering those so thoroughly, and I think that's brought us to the end of the Q&A session. I'm not sure we could have carried on for a while because there inevitably will be the odd question coming through, but it just leaves me to finalise proceedings by thanking all of the registrants for joining the webinar today, and a special thanks to you, Ed, for an excellent talk, and of course to thank Agena Bioscience as well, who sponsored this webinar, so special thanks to them too. This webinar has been recorded, so we will post it on www.thejournalofprecisionmedicine.com. It'll be there for a good 6 months if you're keen to access that in the coming weeks. And all that leaves me to do is wish you all a very pleasant rest of your day, and we look forward to welcoming you all back for another Journal of Precision Medicine webinar very, very soon. So thank you, and bye for now.
Thank you.
Thanks, Ed.